with your knowledge. I may talk over some of you, so I hope I can encompass everybody's level of knowledge, because I'm gonna, after I tell you about why I'm here, how here, we're gonna talk about what is a fly, because I've found that a lot of people don't really know what a fly is. I mean, they hear about caddis flies and lantern flies and all kinds of different names with fly in them, but they don't really know what a true fly is. So I'm going to be talking about true flies and what is that. So I'm not sharing my screen yet, right, Bronwyn?
Not yet, but you're feel free to share whenever you're ready.
Well, I'll just chat a minute. So I want to introduce my wife, Annette, real quick.
Oh, Georgie. He's embarrassing me.
She helped me on this project with the flies.
Hi, everybody.
With the snail-killing flies. We did a survey of snail-killing flies in Maryland, started in 2014. Long-term study, finally published it, 2 years ago in the Maryland Entomologist. Annette is my public relations specialist. When she accompanies me on my field trips, I'm out there swinging and netting around in the muck and the mire, and she's explaining to people what the heck I'm doing. Because I get the weirdest looks, right?
That's true.
Oof. So, which is great. I mean, people should ask. I'm glad when they ask, but she can explain it better than me because I'm busy sucking flies out of my net. So, so that's Annette. So I'm going to be talking about this project we did, but it's only one little aspect of my work with flies. And how I got into this was, I grew up in Rockville, in a suburb where we weren't really near nature, just suburban Rockville. Tons of streets. If you know the Wheaton Woods area, Aspen Hill, some of you may know that area. All I had was a natural curiosity. I read everything. I read books. I didn't know nature. But so when I was in 9th grade, we moved out to Gaithersburg, and we had a place out in the country, with a pond nearby. My brother was in college at the University of Maryland, and he, as one of his electives, he took Entomology 101 by the famous Dr. John Messersmith. Maybe some of you have heard of him. But my brother Warren had to make a collection for this class as a requirement. So he asked me, would I help him? And he gave me, I said, yeah, let's see what this is all about. I went out collecting. I started collecting, and I collected all kinds of insects. I studied everything. I had a huge collection of all insects. And I just found that when I went out into nature, I just wanted to know. I wanted to know the names of things. I look at a plant, like, I don't know the name of this plant. And the insects and the birds and everything. I was just, my curiosity was really lit up. So I studied all kinds of insects, got to know families of pretty much every order. But one day I was collecting down in that pond I mentioned, and I got this speckled fly that was beautiful to me. I said, I'd never known a fly could look like that. And I'm going to show you some pictures of this genus of snail-killing fly. I didn't know what it was. I was able to key it out. I figured out what family it was, but I happen to have a connection with the guys at the Smithsonian. So I went to the University of Maryland. As I grew up, after I grew up, I went to Maryland. I studied entomology. I majored in entomology. I got to know all these guys. And at Maryland, they got me hooked up with the Smithsonian guys, who were world-renowned dipterists, we call them, fly guys. And they were my mentors, and so they helped me get started. I published papers as an undergrad. But getting back to this snail-killing fly, I took it downtown to the world expert. His name is Lloyd Knudsen. And I said, Lloyd, I think I got a new species here. I've never seen anything like it. So he was very nice. He just said, well, let's take a look. And he said, let me show you some things. He showed me about orienting the fly, especially the male genitalia. He said orientations is key. You don't have a new species, but you have this so-and-so species. I'm like, wow, he taught me something. So anyway, because I had published already as an undergraduate, I was invited to join the PhD program, skip my master's. The head of the department said, just go into the PhD program. Don't worry about your master's. I did it for a year, loved it. Some stuff came up in my family and I had to leave the program. Or I could say I chose to leave the program. And I kind of put it behind me because I went into sales and marketing of scientific instruments and scientific equipment. So I was still in science. But so along around in the 90s, I started to get the bug back. The fly bug came back. I'm like, I got to have a project. I got to get back into this, at least on the weekends. So I was still working in my career, but I hooked up with one of the Smithsonian guys. He said, come on down. Let's do some projects. So as of the 90s is when I really got back into this and started collecting again and focusing and writing papers, doing research. I co-authored with this Dr. Wayne Mathis down there. So that really got me back into this. And it was only weekends. I'd go down to Smithsonian, take the metro down to Smithsonian. I spent just tons of time down there. So I think the point is anybody can do this. Anybody can be a scientist. You don't have to have your PhD. You need to have curiosity. So anybody, and I try to, I'm a substitute teacher out at one of the high schools, and I tell students, go out in your backyard and look around. Take a walk in the woods. Look around. See if something makes you ask questions. You can literally become the expert in that thing because nobody else may be looking at it. And I pointed out that, for every living thing, whether it be a bacterium, a worm, fly, bird, fish, there's somebody that knows everything about that thing. There's an expert in the world on that thing, and you can be an expert too. So that's my long story about how I got here. So with that, I'm going to introduce the idea or the question, what actually is a fly? Now, let me, Bronwyn, I got to get that back to sharing my screen. Here we go. I guess you can see my screen. I'm still seeing mine. Does everybody see that?
Yeah, you have to go to your presentation now. Yeah, there you go.
Okay.
If you can hit that that little box on the bottom that says hi?
Hi.
Yeah.
Okay. So what are flies? This is what I ask students when I do. I've done talks for students out at the high school. Most winged insects have 4 wings. So what makes a fly is that they only have 2 wings. Their 4 wings are the wings that they use for flying. The hind wings, they originally had 4 wings way back in evolutionary history, but the hind wings have evolved into these balancing organs called halteres. This is a picture of a crane fly. A lot of these are pretty large. Sometimes they're an inch and a half. People see them in the house flitting around. They go, "Wow, there's a huge mosquito." It's one of the more primitive flies. It's related to mosquitoes, but it's not a mosquito. They don't bite, but there are thousands and thousands of species of them. So this is a picture of one that I think is pretty cool, but it really shows the halters very well. But as you move up the evolutionary scale, the higher flies, you see here this little yellow structure. Yeah, that's the halter. So the bottom line is flies have 2 wings and they have a set of halters. They have 4 life stages: eggs, larvae, or maggots, pupa, and then adult. The larvae are very important to forensic entomologists because they can tell them apart and they can actually figure out how old a corpse is based on the fly larvae that are on them. But when I was mentioning earlier about a lot of things are called flies, mayflies, dragonflies, caddisflies. Well, notice that the word, take mayfly, it's one word with flies in it. So that means it's not a true fly. If it's a separate word like horsefly, that's a fly. A true fly, meaning 2 wings and a halter. So there's a whole gamut. There's, yeah.
Could you maximize the image anyway? Or,
I don't know. How do I do that? Can you see that? Or is it small?
Try this.
I mean, I can see it, but somebody is just asked. Okay, that's better. All right.
No. Thanks to my tech person, Annette.
And publicity more.
Publicity and tech. So before I get to snail-killing flies, I'm just trying to give everyone an idea of where they fit into the scheme of flies, from primitive flies such as the crane fly I showed you, mosquitoes. In between, there's horse flies, flower flies. The advanced ones are like house flies and parasitic tachinid flies, bat flies. I show you some pictures of those. Those are really weird. So the flies that I study and have specialized in for many years are the more advanced. They're called the higher flies or higher Diptera. The word Diptera means it is the name of the order of flies, Diptera. Moths and butterflies, Lepidoptera, Hymenoptera are bees. They all end in optera, meaning wings. So Diptera means 2 wings. Di is 2. Pteron is wings. So true flies are Diptera. And you know there's 17 million flies for every person. And the reason we don't see them, why we're not like deluged with them, is because most of them are in the immature stages. Flies do their thing for the most part in the larval stage. And that's going to lead into the snail-killing flies too, because they do their thing in the larval stage. The adults, really for mating, they do some feeding, except for the biting flies. Now they do their thing as adults, and I think you know about that, mosquitoes and horse flies. So, 17 million for every human, and there's 1,600 new species being described every year by taxonomists. Even fossil, we're still finding fossil flies in amber. So that gives you an idea, I hope, where the snail-killing flies fit in. So let me see. Now, is that large enough? Can that be seen?
Yes, that's perfect. Yep.
All right, I think I covered the bases, unless anybody has any questions about where these little critters fit into the natural world. But this happens to be a family. I already talked about the order Diptera. That's how it's spelled. The Sciomyzids, the family is called Sciomyzidae. My expert friend and I were researching the word scio the other day, and we still haven't figured out quite what it means, except the closest we can get is shadow. So I'm not sure why the original guy named it Sciomyzids, but that's what it's called. The family is called Sciomyzidae. If you know about the binomial naming system, genus and species, well, it goes on up to family, order, family, and then genus and species. But there's also subfamilies and other categories in between. So the Sciomyzids is actually a pretty big family. Not huge, but fairly large, 548 species around the world. If you can see this map, the different regions, the like where we are in the Nearctic, that's what N means, 201 species. Not sure what the second number is. I'm going to ask Bill. P means Palearctic. So actually, if you look at this, you'll see most the species rich areas are more in the temperate zones. The tropics are not as rich. So there are two tribes in North America. Now, the tribe, you take the family Sciomyzoidea, and there are subfamilies. Every family seems to have different subfamilies based on our ability to group things together and based on morphology and also DNA. So there's only one subfamily in North America, the Sciomyzoidea. There's two tribes in that subfamily, the Sciomyzoidea, which are the more primitive ones, and there's 6 different genera that we have around here, and the Tetnosaurini, and these are a little more advanced. They're predators. They're just all outright predators of snails. The Sciomyzoidea are parasitoids. And I'll talk about that in a minute. But it's interesting. The range of sizes of adults here. The one on the left is, we actually have that around here. It's in the genus Tetnosauria. Those get to be about an inch, almost an inch long. And the one on the right is a genus of very tiny ones that get to be maybe, on a good day, 3 millimeters. So when I'm swinging my net around out there, use a device to suck them out of the net and put them into a vial, because they blend in with all the other tiny flies. Actually, what I was going to show you guys, because if anybody's interested in let me see. Is that okay? Can you see that okay? This is just an example of, so is everybody good with seeing that? Bronwyn?
Yep, we're good.
See it.
Can you see it at all?
Yeah, we're good. We can see it.
So this is just an example. I take pictures of the various habitats around Howard County, and where you find these snail-killing flies, and it's really, where are they? They're where the snails are. And where are the snails? Anywhere it's wet. And also there are terrestrial snails that are not necessarily near water, like in dark shady woods. There are, in the grassy areas, there's snails, terrestrial snails, and there is the genus that specializes in attacking those. I'll get to the life cycle of them in a minute, so maybe understand that a little better. But these are the kind of habitats I get to go into. They're pretty little areas. This is an area just down the end of my street. And most of these, by the way, are stormwater in ponds and management structures, and they come in all shapes and sizes. Some are dry, some are wet, some are always wet, some are wet dry. This is one near the Columbia Gym that is always wet, big pond. This is down the end of my street now. Nowadays, that it's totally dry. But this is where I found a new species. Just at the end of my street, you can find a new species. It's amazing what's out there. This is another picture of that little pond at the end of my street. That's what you see, all the green. That's not mold or anything. It's duckweed. It's a little tiny aquatic plant. So this is the kind of habitats where you find them. And like I say, there's I found the big ones down in here, and the little ones I find everywhere. But I have found them including in my backyard. We have a wet spot, and typically in May when we get a lot of rain, it's kind of mucky. Sure enough, snail-killing flies are back there. So they're basically everywhere that is maybe that you can find a snail. Let me see. So that's the size range. This is just a picture of the different genera. Now there's genera and then these. A genus is a group of like species or closely related species, and that's determined by the person that's studying. They make that determination. Sometimes we're wrong. We put things in the wrong genus, only to find out later it's actually more related to this other guy over here. I like this picture because it shows the range, pretty little colors of them, and why I got to love them. I mean, some of these are just really colorful, and these are all found around here. So this is the genus Verbelia. This is in the more primitive tribe. These are parasitoids, meaning, well, I'll get to the life cycle in a minute, but they actually, the adults lay eggs on a snail, and these attack them. And these are tiny. Most of them are tiny. This was my favorite genus. This is what got me into flies, Dictya. Dictya, Dictya. Very common family, genus. It's the most common genus in Howard County. One species, Texensis, is the most common species. But I thought it was beautiful. Their mottled appearance, and it's amazing. In this particular genus, you cannot tell the adults apart as far as the species. You must do what we call dissect the male genitalia. The male genitalia in flies can be very complex, and they are usually very species specific in terms of their structures and the morphology of those structures. And in my early days, when I first discovered that we could do these dissections and figure out what's what, I really became just enthralled. Like, wow, they look identical. This could possibly be 3 different species, and they're identical. Externally. And this is my guy, Dictya. They got that spot in their face looking right at you there. So these are basically, if you go swing a net around, you're bound to find one of these guys. This is my second favorite genus in Howard County, Euthicera. Another beautiful mottled fly. And these are large too. And these attack terrestrial snails. If you go in the woods in a like dark, shady area, moist woods, and swing your net through the grasses, and ferns too, by the way. Out at Glenelg High School, they have a woods next to us that's full of ferns, and I collect these all the time out there. So there must be a terrestrial snails in those ferns, and these guys are after them. Just a few of the genera. They're just this genus Linnea. They can be found in whales. I collected these out, oddly enough, in Marriottsville at the what do you call it? The landfill. They have a picnic area in the entrance to the landfill. No pond around, just grass. I collected these Limnea in there. So they're finding things that we have no idea as far as snails. And actually, if you look at the antennae of this family, they're all really erect, and they must be full of sensory hairs. And they are. We actually have scanning electron microscope pictures of these antennae. There's another Limnea. Little jazz music. Oh, and I want to acknowledge my friend and colleague, Bill Murphy. He loaned me this presentation. I said, Bill, you got a presentation? Instead of me reinventing the wheel, he said, sure. So I just want to acknowledge that this is his presentation. This genus, Piscillagrapha, I'm still looking for these guys. Supposedly they have a very short flight period, like I think early spring, and maybe just a couple of weeks. So to get one is quite a score, and they are pretty. So that's one of my boxes to check off in life, is to collect some of these guys. This is the genus Cepodon. These can get really big, and we know the most about these guys in terms of their biology. I guess maybe they're more easily reared or something, but people who study life histories know a lot about this genus. Can you see my cursor? So if I point something out?
Yes, we can. Yes, we can.
Again, the antennae. I mean, I don't have, scientific data on how they find snails, but all of the species in this family, first of all, they stand on leaves and of grass and emergent vegetation in ponds. They're always facing downward, and I swear they're with these antennae. They can smell a snail. I mean, to me, again, the endless diversity and interest in nature. How do they do that? I think this one moves too. Yep. Typical fly posture, cleaning my front legs and my feet. Looking around, waving his antennae, detecting snails. Another pretty little model guy. I've collected plenty of these guys. I should have had him put one of those fly songs in for these instead of the little jazz, learning to fly or something. This is what they look like when they're mating. Typically, the male is on top. And they are very active maters. I mean, I've seen, especially the genus Dictya, in the vial that I capture them in, they are going about mating. So they are very prolific. Little loving jazz music. The male is putting his feet on the forehead of the female. Not sure why. Maybe it calms her down. Don't know. So here's how they live. The female lays eggs. In the one tribe, the parasitoids, they lay eggs directly on a snail. So the female finds a snail, pops a little egg on the snail. And the predatory tribe, Tetanosaurinae, they're laying eggs on the vegetation around wet areas, and I assume even like in my backyard on the grass, so that when the larvae come out, they have to find a snail. But the larvae come out within 2 to 5 days. And of course, fly larvae are called maggots typically. And so then they go looking for snails. And these are and I don't know if anybody here knows about snails. I'm not really knowledgeable on snails, but these two genera are air breathing, found in our local ponds. They have lungs. They're called pulmonate snails. And they come in all sizes. You can see the tiny little black ones. I have seen these around. So, and also, interestingly, the maggots, as they grow, they pick larger and larger snails. So I actually found one site, stormwater management. It was more or less a mud flat. It was full of little black snails. And plenty of snail-killing flies were there. It's another genus, Planorbella. So just a little side note, snails are everywhere and snail-killing flies are everywhere. If you go out in the woods, go through the muck and the mire, you're going to find, if you really look for it, you got to look for these snails. It's another genus. These are terrestrial. So they don't necessarily need to be in ponds. And slugs. Slugs are also mollusks. They get fed on by these flies. So the larva just goes in, and this larva actually probably has just finished this snail, because look how big he is. He probably started small. This is a scanning electron microscope photo of the back end of a snail-killing fly larva. Those strands help them float and breathe. This is how we tell them apart. The only hard parts on a larva or a maggot are the mouth parts. And these are used, interesting enough, in forensically important flies. They use these to determine what species they are. They can tell how old a corpse is.
Snails belonging to various species of Limnea in different parts of the world are required for development of the liver fluke parasite of sheep, cattle, and man. Control of these host snails would curtail transmission of this parasite and limit the spread of the serious disease it causes. The tropical American snail, Biomphalaria glabrata, is easily killed by Sciomyzoidea larvae. Other species of this genus that transmit schistosomiasis in Asia, Africa, and South America also are quite vulnerable to their attacks. The cloud of reddish pigment that will appear right beneath this snail's aperture shows how quickly a larva may rupture the blood sinuses and cause the snail to hemorrhage. Aquatic snail-killing Sciomyzoidea larvae feed rapidly and leave their food snails as soon as their hunger is satisfied. When hungry again, they kill another snail, and each larva may kill as many as 24 snails. They never attack anything except snails, even when they are starving. Adult Sciomyzoidea have a broad sponge-like proboscis, which they use for sucking up droplets of dew and flower nectar. They are not attracted to man or domestic animals and are rarely seen except in the marshes and swamps where their larvae are breeding. They also feed avidly on snails, both living and dead. Since they don't injure living snails, they probably just ingest the proteinaceous mucus that the snails secrete. Dead snails, which the larvae have killed but not completely eaten, probably make up an important part of their natural diet.
So after the larva, they pupate, which means they form, they kind of contract and form a hard outer coating. And typically they're in the pupal stage 10 days or more. In the case of the parasitoid tribe, they're typically in the snail, so they kind of will form or conform to the sides of the walls of the snail shell. So they can vary in shape that way. The predatory species are typically found floating on ponds, and they look like little rice grains. And a number of these genera, like the genus, my favorite, Dictea, and some of the others, they're actually overwintering right now as pupae. And also, and some of them are in the advanced pupal stage called the pharate adults. So they're basically adults ready to pop out. So we found in our survey, because I went out all the time. I didn't, I went out every month to see what I could find, and especially on warm days, which we know around in Maryland, we get warm days. We'll get days in the 60s for 2 or 3 days in a row sometimes. Not so much in January, but come February, we're going to see a couple of days here and there that are in the 60s, even 70s. You can go out. Typically, if it's 2 days, by the second day, you can actually go out and find flies that are on the wing. Not just snail-killing flies, but all kinds of flies. So, and I'm sure that applies to insects in general. I mean, they're not just warm weather beasts, and there are some actually primitive flies that are found on snow in the old world. So right now in ponds, there are all these pupae floating around and probably resting on the mud, getting ready to pop out. So the video was focused on these bloodborne or snail-borne diseases. Schistosomiasis is one of the big ones, and there's several others, as you can see here. Back in the 60s, I think, my mentor, Lloyd Knutson, was at Cornell, and they got involved with determining the life histories of the species in this family. And they found that some of them are actually useful in controlling snails in tropical areas where these diseases are a problem. And the diseases are not that uncommon. I'm not going to go into the life cycle too much, but it's kind of a complex life cycle. You see here, this is the egg stage of the disease, or the disease organism. They hatch in the water. They go into snails. So they're in snails, and they form and become adults, or free swimming forms in the snails, and come out into the water, and they can penetrate a human skin when you're in the water. Which, as we know, in tropical areas, people use the natural water sources for bathing and drinking and laundry. So they're in the water quite a bit. They can pick up these disease organisms, and they end up in the liver, and they can cause some pretty nasty looking symptoms. And so they found that they can use these Sciomyzids for biological control in the tropics. So they do serve an important function for man. There's Lloyd Knutson wrote with Jean-Claude Vallat, a French researcher, a book on Sciomyzids in 2011. There's a whole book on them. And this is my colleague, William Murphy, and two of my mentors. Now, Lloyd Knutson has passed away. He's deceased now. This is the major work that they put out on the Delmarva Sciomyzids that I use as a manual in determining and understanding the life histories. They've also created a website for helping to determine the genera and species of Sciomyzids. This is just a picture of some of the pupae that have been found just floating in the water or on snails. So that's that part of the talk. That's it. That's the talk, really. Murph 3000. That's my buddy Murphy. Feel free to write to him if you want more information, or perhaps a copy of this. Let me see.
All right, you can just unshare, George. Just hit stop share.
Okay, I got it. There it goes.
There you go. All right, thank you so much. That was fascinating. We have some questions in the chat box.
I hope it didn't bore you.
I don't think so. I think we're real excited about learning about these little guys. And the first question came from, and if you want to ask George a question directly, just raise your hand. I'll call on you, and you can unmute, or you can put it in the chat box. Richard has a very a question about speciation, and this could probably be a 12-hour discussion in and of itself. But what is your Y-O-U-R, your George's definition of a species? Is it as simple as the reproductivity isolate, or what properties are you looking at and record to justify designating a new species?
That is the core and the crux of taxonomy. And that, depending on the organisms you're working with, the definition of species can vary. But as far as flies, it is a complex question, because we're finding that, well, traditionally we use morphology, but sometimes we find that morphology can be variable within certain limits. What do I use? typically for flies, we use male and female genitalia. We use external morphological features. So I may look, typically you're looking at thousands of specimens. And when you keep looking at them over and over, there's not a whole lot of variability. So that the assumption is, and I emphasize assumption, is that they are the same species if they share the same morphology. And that assumption and hypothesis has worked out by and large for us taxonomists. It seems to be hold true for most insects. There's another definition, and that is whether or not individuals can mate and produce fertile offspring. But that information is not always known. I mean, we're finding new species all the time. We don't know much about their life histories. So I use, and most of us taxonomists, we use morphology as evidence that something is a species. That species A is different from species B, and we give them two different names. We have found that the male genitalia have the least variation. Maybe has something to do with being able to mate with just the right female. Maybe it's a lock and key kind of arrangement that's got to be specific. But we found that in the genitalia are the most reliable indicator or evidence that something is a different species. So it requires a lot of dissection, and you're going through studying these genitalia, and all of a sudden, it pops right out at you. This is different. The structure is different. The structure is round, and the other one is pointed. So I hope that answers your question without too much verbosity. It's a tough question, actually, and it depends on what you're studying.
Yeah,
Great question.
It's a question. All right, Alan, and you mentioned earlier in the talk the variation in sizes, and can you say how big is big when you say that these are big ones and these are small ones?
Yeah, let me see. Where's my paper? This is our paper that came out in the Maryland Entomologist. Let's see where the survey of the snail-killing flies at Howard County came out in 2023. It's basically adults range in size from 1.7 millimeters, so even smaller than 2 millimeters. I mentioned 3 millimeters, so they're even smaller than that, up to about 13 millimeters. So basically half an inch. In Howard County, we found that the range was more like 2.4 for Forbelia nana to about 7 millimeters for the genus Tetanoceros. Some of those get a little larger than that, perhaps. So around 2 millimeters up to even as high as half an inch.
All right, thank you. Richard also wants to know what the difference is between parasite and a parasitoid.
Yeah, a parasite. Now, this is a tough one for me to answer because I don't deal a lot with parasitism, so I tend to forget the difference. But parasites tend to oftentimes will kill their host. Parasitoids feed on them, often without killing them. That's my best answer because there are different forms of both. I would suggest that you do a little Wikipedia on it. I've done that. They have a really nice explanation, and some of the various university websites can give you examples of parasitoid versus parasite.
All right, sounds like a good topic for another talk.
Yeah.
Jackie asks if the hairy appendages help them to float to keep them from drowning. What do the maggots do when the snails submerge?
Good question. I think they stick with them. they're just inside. That's a good question. I don't know. I got to ask Bill Murphy that question. I never thought of that one. Thank
All
you.
right, Jackie, we got a good question here. We're going to find an answer. Let's see. Megan asks, there are a lot of invasive slugs here, as well as non-native snails. Do you know anything about the Sciomyzids attacking the invasive species? And then Timothy adds on to Megan's question, are any of the ones that you've encountered non-native species, or are they all native flies?
Yeah. So far, unfortunately, just native. We're not. I haven't found, and I don't know of any invasive species of Sciomyzids. Interesting question. Never thought about that, but yeah. Everything that I've seen and everything that I know of, and in working with my colleagues, yeah, they're pretty much limited. They don't really spread as much. There are probably a few introductions, but they're not like some insects that will get in grains or plants that get shipped all over the world. They tend to be located, in their little habitat, their marsh or wet grassy areas. So they're not typically attached to things that get shipped around, and so that we find them in places where they shouldn't be. So yeah, actually that brings up a good point. Is we find new species. I say all the time, but I've described 40 new species so far of beach flies and grass flies. I haven't personally described any new Sciomyzids, but with intensive collecting in one area, you tend to find things that you might not find otherwise, and that's how I found this new species. I didn't know it was new, but I sent it to Bill Murphy, and he said, you have a new species here. I'm just getting ready to describe. He had a paper getting ready to be published, and he said, you found it in Howard County. And so he described it, and he named it. But since then, since the first time I found one, I found 13 or 14 more. And he's blown away. He says, obviously, Howard County is like the global center for this species. He named it after his brother. But it's this intensive collecting, and that's why I did the survey, because most collectors, you go to a site one or two times, and you don't really know what's going on there over the course of the year. And I know I'm not quite answering your question, but another interesting thing I found by going back to this one particular site nearby, I went there all year long. I found that with the genus Dictea, there was a temporal or a time-based separation of the species. In the early spring, March and April, I find 3 or 4 species. And then along comes, June, July, I find 3 or 4 different ones. And so they overlap. And along in about the fall, this one falls away, that one shows up. And there are some species like Texensis that are basically, we collect them all the time. So there's a difference. We think they may be dividing the habitat based on when they're there. What was the other part of your question? I got off track.
The other part was, are they eating any of the invasive snails or slugs?
Probably not to an extent that makes a difference. They're just not, when you find Sciomyzids, you find a few, 2 or 3, maybe as much as a dozen, but they're not like thick with them, so they're making a huge impact on the snail or the slug population. But that's a guess. Don't have any data on that. They may be having an effect, and maybe that's why we don't see more slugs.
Jackie wants to know, what type of netting do you use to catch the flies?
I use it's a very fine mesh net. it's the finest mesh that you can buy, so that the little 1 and 2 millimeter flies can't escape it. But it's a long handled net with a, I don't know, maybe what, a 1.5 foot diameter ring on it, and maybe a 2 or 3 foot net, and it's cone shaped, so that when I swing it through the grasses, I can capture the flies down on the pointed end of the net, so that's sticking up, and I can go in with an aspirator and suck out the little flies. So it's a very fine mesh, commonly can easily be purchased. I think Amazon has them, and there's a new company, Ecology Supplies, that has them. But the key is the fine mesh. Like the folks that collect butterflies and the larger things, they use a much larger mesh. But I'd lose everything if I use that, except for the Sciomyzoids.
And how do you suck it out with the aspirator and you're not swallowing them?
Yeah, so the aspirator, it's got 2 tubes on it. One tube is to suck in the flies, and that goes into a vial. Out of the other side of the vial is a tube that goes into a long tube that I put in my mouth. But on the base of that tube inside the vial, I use old netting material and cut a square out, and then sticking it through a cork. It's a cork on top of the vial that both of these tubes fit through. So I put that fine mesh on the bottom of that tube, shove the tube inside there. So I now have a barrier, a net, a filter that keeps me from aspirating the flies, which is, I've done that, and that is not pleasant.
Dave wants to know, can you give advice on how to rear Sciomyzoid larvae as pupa?
Yeah, I haven't done it myself because I don't really have the resources here in my house for it. But I know that's a big thing. The guys at Cornell have done this, and other universities and museums have done this. But what you do is you go find some snails, and you can even have an aquarium or terrarium, just a wet terrarium with a fine mesh over the top, and have your snails in there. And I don't know what you feed them or anything, but probably they'll be okay eating algae. But just find snails, collect as many snails as you can, put them in a terrarium so that anything that comes out of them gets captured. Now the researchers typically don't use a terrarium; they use small vials or bottles so that they can isolate one snail at a time. If you have 2 and you get 2 different things, then you got too many variables and you don't know what's what. So it depends on what you're looking for. If you're just kind of fascinated by stuff emerging, just go down to your local pond and flop around in there and find some snails and put them in a vial. Keep it wet, like wet sand. I can find more on that, and I'm sure there's more information on how to rear, not rear snails, but keep them alive until something comes out of them.
Would you go and collect the pupae that's in the water and then and see what emerges out of them?
I haven't myself. I've been focused on the adults. My colleague Bill Murphy has done this, and he's reared them out. And he says you can find in the wintertime, you find them all the time. The pupae floating on the surface of a pond. You got to get down on your knees and really look closely. But he has done that and reared them out. It's fascinating. But I have not done that myself. I don't really have a place to do that. But apparently they look like little rice grains floating on the surface of a pond.
I know what Dave's going to be doing tomorrow.
Yeah.
And Jackie, it says, is there an economic value to this
To the survey?
I
Or to
think
the
so. Study or what you just published or studying these insects.
I wouldn't say it's economic so much as scientific, just understanding the distribution of them, and the fact that no one's ever gone back and collected over several years in one site. It's scientific data. It's distribution, biology, just adds to the general knowledge of nature of these particular flies. Yeah, they don't have an economic importance per se, but they have a medical importance in some parts of the world. But the reason we do surveys is just to find out what's here, basic scientific, understanding.
Dave says, do they feed on any other mollusks like pea clams?
Yes, there's some that feed on fingernail clams. That's a good point because I love salt marshes. I've just published a paper last July on several species of grass flies that are only found in salt marshes. But the snails in salt marshes, there's 2 species of Dictya that only occur in salt marshes. So apparently they go after various mollusks in salt marshes, and there's a species, I don't think it's Dictya, but that attacks fingernail clams and razor clams. Good one. Yeah, like any mollusk is like prey for these guys.
And you focused on Howard County. Has there been any other detailed studies in any of the other counties of Maryland for these?
No, and that's the problem. I mean, there's probably a lot more to be discovered if someone would just do what I did, and especially in like Western Maryland, in the mountains, or Eastern Shore habitats, the Piedmont, salt marshes. I've done a lot in salt marshes, and I've only found these 2 species, but so much work needs to be done. I mean, somebody could really take this on in any county, and especially going south, Prince George's or Southern Maryland, because I don't think we know much about what's down there in the closer to the warmer south. There is a species of Dictya that I think this is its northern range, Howard County. So yeah, much work needs to be done. Now,
well,
there's been surveys done all around the world in different areas. I know Murphy has done one for Indiana and various states and counties that he has gone into. So yeah, these faunal surveys are done quite a bit. You can see them in all types of organisms. The people just want to find out, where are they? What do they feed on? What do we collect them on? Time of day, time of year, just general knowledge of the natural world. We just want to know.
We certainly do. We certainly do. Does anybody have any other questions for George tonight on our snail-eating flies?
And anybody ever heard of them before? Probably not.
I had not, no. But I do now, and I know what to look for, and I'm gonna be looking for them.
If you want to delve into flies, I would encourage anybody to go look up bat flies. Family Strepsilidae, but you could just do a search on bat flies and look at the pictures of them. They do not look like flies. They look like spiders. The head is reduced. Why do we know they're flies? They have halters. But they are really cool. I was going to show you guys some pictures of them, but look them up for fun. The fly diversity is unbelievable. So I've just bitten off a little piece of the whole fly picture with these snail-killing flies.
Well, now we're all going to fly off from this presentation and go look for our flies and enjoy it. So thank you so much, George, for sharing your passion, your knowledge, your expertise, your flies, your stories with us. It's been a presentation.
I hope everybody enjoyed it.
We did, and I maybe we can go out collecting if you want. I'm sure some of these folks here would love to go out collecting with you and learn how you do it, and maybe do a tutorial so they can go and do it back at in their own little parts of the world as well. So we can continue
That'd be a
to
blast.
share that.
That would be fun. I mean, we could collect them and show you how to pin them and identify them and stuff like that. A lot of people are into photography now, which is good. There's less collecting going on. The stuff that I'm studying, you can't identify them on pictures because they're so small, and the differences between species are so fine. Some of them is just the pattern of hairs on the back. So I can't use photography so much, but there is a movement to get away from collecting and killing things out there. But there are some things that we just have to, because there's no other way to know about them. So just throw that in, because there is some sensitivity, and I totally understand it, about taking things out of nature. In fact, you got to get permit everywhere now. In order to go collecting, most places you need permits, which is good.
That's a good question. What kind of macro lens were you using there for some of those pictures? Or maybe it was your friend who was.
Yeah, I think they were online somewhere. I think he found them. I am not a photographer per se. I take pictures through my microscope. I have a 4D camera through my microscope, and I can take pictures of really tiny flies that I'm working on. I don't do photography, and to me, that is so amazing that they can even do that. The pictures that they can take of little things on a blade of grass. But that's not my specialty. Good question.
All right. Well, thank you everybody for joining us tonight. Next week we have invasive earthworms and how they affect the forest soil composition and ecology. That should be fascinating as well. Come back and join us for that, or anything else, and come visit me at the museum and say hello and goodbye to the woolly mammoth. And George, we'll be talking on how to do a little field exploration with you as the temperatures warm up. That would be awesome extension. Everybody stay well, stay curious, stay outside, stay warm.
Yeah. Bye-bye.